And all right, so good morning, everyone. So today we have our guest speaker, Dr. Maria Contel, and I'm going to actually have the graduate student Isabel Rodriguez provide the introduction.
Good afternoon, everyone. So Dr. Maria Contel received her combined bachelor's and master's degree in chemical sciences at the University of Zaragoza, Spain, and received her PhD in chemistry at the Public University of Navarra under the mentorship of Professor Mariano Laguna and Julian Garrido, where her research was based on gold complexes as precursors to polynuclear complexes. Dr. Contel has 2 postdoctoral experiences, one from the Research School of Chemistry at the Australian National University, where she performed research under the supervision of Professor Martine A. Bennett. Her second postdoctoral experience was from the Dubai Institute at the University of Utrecht in the Netherlands, and she performed research under the supervision of Professor Gerard van Cotten. She was a principal investigator at the Spanish Ministry of Science and Education for 5 years, located at the University of Zaragoza, Spain. There she researched compounds of groups 10 and 11 metals as recyclable catalysis for carbon-carbon and carbon heteroatom bond formations. In 2006, she began as an assistant professor in the chemistry department at CUNY Brooklyn College, and is now a full professor of inorganic chemistry. She has also served as chairperson from 2017 to 2020 at CUNY Brooklyn College. In August 2020, Dr. Contel began as a director of Brooklyn College Cancer Center for Community Outreach, Research, and Education, where she was a co-founder of. Her current research is based on medicinal inorganic chemistry and has expertise in the development of heterometallic compounds for anti-cancer applications. She's a very prolific researcher with more than 75 publications, an H-index of 36, and she has 3 patents as well. She is funded by the NIH, American Cancer Society, and the Gray Foundation. She has many recognitions. Her most are the 2022 Outstanding Faculty Science Award from Brooklyn College. She was elected vice chair for the 2024 Metals in Medicine Gordon Conference. She has mentored 7 PhD students, 3 master students, 15 postdocs, and more than 40 bachelor students and high school students, with 80% of researchers being female or underrepresented students. And with this introduction, I would like to turn it over to Dr. Maria Contel.
Thank you, Israel. That was a very, very kind introduction. Thanks a lot. I had forgotten some of the things I did in the past. So let me share my screen. And well, let me first say buenos días a todos los estudiantes de la Universidad de Puerto Rico del curso de Metales en Medicina. Voy a dar esta charla en inglés, pero si tienen cualquier pregunta, por favor, pueden hablar mientras estoy dando esta charla o pueden escribir en el chat y el profesor Arthur Tinoco me hará él las preguntas. Sorry for that little thing, Aaron, because Aaron doesn't speak Spanish, I think. I just wanted to let everybody know you can also speak in Spanish. Okay, let me share my screen now. And can everybody see?
Yes.
Okay, so, okay, let me see. Great. So today, I'm going to be talking about one of the multiple projects that we have in my laboratory. This particular one is about compounds that have more than one metal, more than one different metal, and the application that these complexes can have as anti-cancer chemotherapeutics. So when I started on this field, there was a lot about, is it really true that 2 metals are more than the sum of its parts or not? And I will try to answer this question. These pictures here are from Brooklyn Bridge in New York. This is what we see from the Brooklyn side. The picture in the middle is from Prospect Park in Brooklyn, a beautiful park. And the other picture is Brooklyn College, which is one of the 23 colleges at CUNY. And we have a doctoral program that is in consortia with different colleges and managed through the Graduate Center. And I'm a faculty member of chemistry, biochemistry and biology programs. So if anybody would be interested, we have a lot of opportunities and different grants from NSF and from NIH, so that you could be funded, especially if you are a US citizen. But we have very different possibilities. Anybody interested, please contact me. So let me see, because, okay. So I want to talk a little bit about the advantages of metal compounds with respect to other drugs. I know that you are taking this course and you will be very familiar, but I just wanted to go a little bit about this. So some of the advantages is that there is a huge range of coordination numbers and geometries for metals. And also we can access different redox states that can be very convenient in terms of oxidation and reduction. We can tune the thermodynamics and kinetics by substituting ligands in these complexes. And then we can organize organic ligands around the metal, and we can organize them tridimensionally in order to target the receptors. And of course, we have a wide structural diversity, so we can choose in a rational way. Sorry. I just having a little bit of trouble to just move. Okay. So this is a picture of very, very general of a coordinated complex for those not so familiar. So you have a metal in the center and you have different what we call ligands that are coordinated. These compounds can be neutral, can be cationic or anionic, and you are going to have a counterion if that's the case. And in general, the metal atom is the Lewis acid and the ligands are going to be the Lewis bases. Sorry. Give me, I don't know why I'm having a little bit of trouble. I think it's, sorry. Okay, so in solution, we are going to have an inner coordination sphere, and then we are going to have also the molecules of solvent. And this is also important when we speak about the stability, which is very important when we talk about metal complexes, or in general, when we talk about metal drugs. However, metal drugs are most of the part prodrugs that lose or exchange ligands, and then to give you the biological active species. So platinum compounds have been used as chemotherapeutics since the 70s with the introduction of cisplatin. Then there were some other compounds like carboplatin and oxaliplatin, similar, but with improved pharmacological profile, and also some other derivatives that were approved in China, in Japan, or South Korea. And these compounds have been the established treatment for ovarian cancer, testicular cancer, head and neck cancer, and small lung cancer during the last 30 years, either alone or in combination therapy. And on many occasions and on different countries of the world, they are given even for different cancers. So the mechanism of action of cisplatin that I imagine that in this class you are very familiar with is that you can have the molecule entering the cell either in a passive or active way, and then this molecule is going to be hydrolyzed and enter the nuclei, or else it's going to react with sulfur donors in the cell and also will enter the nuclei, and then it's going to be damaging DNA because it's going to be attaching in a covalent way. And if there is no DNA repair, it's going to produce apoptosis. However, only around 5% of the platinum goes into the cells and the nuclei. So that is not very good. There are some events for which the cancer cells become resistant to platinum-based treatments. So one of them is the possibility of the decreased drug accumulation or increased drug flux. Then there can be a detoxification of the drug because in the cell you have molecules that contain thiols, and that can be a potential pathway to avoid entering in the nuclei and or doing some other things to damage the cancer cells. They can be enhanced repair and increased tolerance to DNA damage by the cells. Cancer cells are quite clever for this. And then there can be also changes in molecular pathways that are involved in the regulation of cell survival or cell death. So all in all, there are some disadvantages and obviously clinical problems that are associated to the use of cisplatin and platinum-based compounds. One is this acquired or intrinsic resistance that these cancer cells display. The other one, very important, is that the activity is limited to very specific cancers. Otherwise, it's going to be a lot of toxicity because cisplatin is not so active in many cancers, even if given to patients for those cancers. And then there is the high toxicity due to the fact that it interacts with DNA, not only of the cancer cells, but of the normal cells. And this is leading to undesired secondary effects. So there has been a lot of studies on the strategies to improve the pharmacological profile and bioavailability for cisplatin and other derivatives. So, and as I said, it still is used in to treat 40 to 80% of cancers all over the world. So there has been a lot of studies in understanding resistance and transport mechanisms to make things better and to design some other platinum compounds. There is a lot of research in what is called unconventional platinum-based compounds that can be, instead of having the ligands in cis, is having the ligands in trans, or having compounds with many different platinum centers, or the use of platinum-4 derivatives, which I will uncover a little later. You can also employ targeting vectors like some peptides that can be, and then you are going to direct them to a specific tumors. Of course, nanotechnology is a big deal with different nanovehicles that have been used for platinum compounds. And of course, another strategy, which is different, is not to use platinum derivatives, but to use other metals, which is something that we've been doing in my group. So these other metals have different modes of action that are not similar to those of cisplatin in most cases. Many of these other metals display weak interaction or no interaction with DNA. Sometimes you have the inhibition of mitochondrial enzymes, or you can inhibit proteases or specific protein kinases, depending. And that has made to even revise the mode of action of cisplatin, which is much more complex, but only affecting the DNA, as I explained. I really recommend this review in chemistry by Boros, Dyson, and Gasser, because it's a really nice review on the different modes of action of most important metallic compounds. So these are the compounds that are inorganic compounds that have been in clinical trials dealing with cancer. So the first derivative is titanosin dichloride, which is a titanium-4 compound with 2 cyclopentadienyl ligands. And this compound entered clinical trials for renal cancer and other types of cancer too. And it was discontinued, but it was discontinued for a number of reasons. And one of the main reasons we believe is because of the low solubility of the titanosin dichloride and the possibility for giving aggregation because of the hydroxides and the oxo species form. So there was a lot of interest in titanium compounds after, much more improved. And I think in the group of Arthur Tinoco, you have many examples of improved derivatives. Then there are this family of compounds of NAMI-A, which is a ruthenium-3 derivative with different counter ions. And they were in clinical trials, even phase 2 for colorectal cancer. And the results were quite good, but not as good as to qualify to go to phase 3. But currently, the derivative BOL100, which is a variation, and it contains a sodium and much better solubility, has been developed by the company BOL100 Therapeutics and is in different phases. Right now, it's in phase 2 to treat different solid tumors in combination therapy with FOLFOX. And that seems to be going very well. Then we have auranofin, which is a gold-1 compound with a 3-ethylphosphine and a thiolate. And this compound was approved by FDA for the treatment of rheumatoid arthritis. And more recently, it's been explored as a potential antimicrobial, antiparasitic, and also anti-cancer agent, and has entered some clinical trials, which are in phase 1. And then lastly, this derivative of ruthenium-2, which is used in photodynamic therapy and is currently entering phase 3 for the treatment in photodynamic therapy of bladder cancer. So this is where it is in terms of the clinical trials. In terms of gold compounds as cancer agents, you have sort of 3 main families. In the family of gold-1 derivatives, you have compounds that are phosphanes or phosphines, like auranofin and thiolates, or phosphines and some other ligands that are charged. And then you have the whole family of gold-1 derivatives that incorporate N-heterocyclic carbines, and they can be 2 or they can be 1 and a chloride. There is a lot of variations. In terms of gold-1, these have been the most important derivatives. For gold-3, there has been different compounds. One of the issues with gold-3 is that it reduces to gold-1 and gold-0 quickly, so they are not so stable in biological fluids, so you always have to stabilize them. One strategy is to use chelating ligands, so these type of porphyrins, like the one shown there for gold 1A, or you can also stabilize them by using cyclometalated compounds. There are many cyclometalated compounds that have been used. This is just some examples from my own laboratory. And then when you stabilize them, they can have really powerful anti-cancer activity. The way these compounds work is they have a mode of action different than that of platinum-based derivatives. One of the main targets is thioredoxin reductase, and they are highly cytotoxic, and in some cases, not in all, in some cases they are quite selective. And there has been examples of the relevant in vivo efficacy. There is the effects of immunogenic cell. There has been recently a lot of interest in what these compounds do to the immune system and the combined effects as anti-cancer. And there has been some recent clinical trials with auranofin, which in my opinion is not the best possible compound, but I think it's reviving all this field. And I also recommend this excellent recent review in Adjuvante Chemie by Cassini and co-workers, and you will see for gold and also in terms of the nanotechnology. And in terms of nanovehicles for gold anti-cancer agents, I also encourage you to check my website because we have some very nice recent examples of using antibody drug conjugates and of using peptide that are enzyme responsive. But before I move to what we are going to speak today, I just want to go very quickly about the hallmarks of cancer. So these are sets of metabolic and signaling characteristics that are unique to cancer cells. And then there are different ones and for which the cancer cells are able to do everything that they do because they can evade growth suppressors, they can avoid the destruction by the immune system, they can replicate, they can promote inflammation, they can activate invasion and metastasis, induce angiogenesis, induce genome instability. They can resist cell death. They regulate cellular energetics. They can do all those things, and that's what it makes such a complicated disease. And in terms of metal-based compounds, we know at least how cisplatin acts on these hallmarks. It acts on the evading the growth suppressors and resisting cell death and the inactivation of DNA repair genes when it works, and it works well. That's what it does. For example, the ruthenium derivative NAMI-A acts more into inhibiting invasion and metastasis and also activating specific cellular oncogenes. And gold auranofin, for example, works in terms of the inflammation, inhibits angiogenesis, and also inhibits invasion and metastasis. So we will be speaking about some of these things today. I want to use also the chance to tell you a little bit how drugs are developed, how in preclinical phase, of course. And then you start by a design of the drug, and you can include computational studies. More recently, this has become very popular. And after you have designed your drug, you synthesize it and characterize it fully. And it is very important. And people sometimes wait a long time to study the stability of the compounds over time. And you can study them. If they are not stable in water, you are going to study them in DMSO. And in DMSO mixtures, if they are soluble in water, but then you have to move to serum, you have to move to human plasma. And that is important because the stability, or at least knowing if your drug is a prodrug and what the biological active species are, is very relevant. So in the past, people spent a lot of time doing interactions with isolated DNA and specific enzymes, and this is still okay, and we still do it. But I think it's very important to go into in vitro cellular studies. And in these studies, we can see cell viability, how many cells, cancer cells, these compounds kill, if they are selective, if they don't kill non-cancerous cells, then the type of cell death that they produce and the type of pathways that are involved, and then which specific enzymes are inhibited, if any, and then you can do many more detailed mechanistic studies by using proteomics, and then genetic and epigenetic analysis, ideally. Then you want to go to a study in vivo and in mice, and then you want to do what is called efficacy in vivo trials to see what happens to those tumors. I'm going to be explaining a little bit more in this talk. And then you want to also do studies in pharmacokinetics. You want to study the pathology of the different tissues and the blood work. And then you want to do a little bit more of the toxicology and pharmacology side. And again, you want to do mechanistic studies of what happens in those tissues, in the tumor tissue, with respect to other tissues when the mice have been treated. And after that, you want to really improve the formulation and delivery strategies of that particular drug that has been good. And then is when you are going to go and try to scale up the synthesis, and then going to scale up the dosage for human use. And in this point is when you can go to clinical trials, which is a topic for another day. So before I move on, any questions by anybody in the audience? I guess
No
not.
question at the moment. Yeah.
Okay, okay. And again, I'm aware that you may have known some of these, but I'd rather be safe than sorry. So I'm going to talk about heterometallic compounds in medicinal chemistry, in which you use the possibility of having two different compounds. And what can be the advantages? Well, each different metal with a different mechanism can interact with different biological targets, what is called a cooperative effect. Not only this, but also, or in addition, you are going to change the chemical-physical properties of the resulting molecule by the addition of a second metallic fragment. And that can be also helpful. And I think we're going to have examples of both effects in some of the compounds that are going to describe. If you want to learn a little bit more about this, we wrote a chapter in this book in 2019, and which was pretty thorough. And more recently, with one PhD student from Puerto Rico, Javier López Hernández, we just did mini review in which we have really gone further in examples that were studied in vivo in mice. Okay. So I'm going to start by talking about heterometallic compounds that contain titanosine and gold. I'm going to talk about goal 1 with titanosin. I'm going to talk about goal 1 with ruthenium derivatives, and finally about goal 1 with platinum 4 derivatives. So all of them have gold as metals, and I already commented on the anti-cancer properties that the gold moiety may display. So this is a project that started in 2011 and then again 2014. So we've been a few years with this project. Different people have been involved, postdocs, and also visiting doctoral students, undergraduate students. And when we started, we really started looking at the molecule of titanosin dichloride that I said before was active in renal cancer, had been a study in renal cancer in clinical trials. And then we attached phosphines into the cyclopentadienyl. With the idea of having that potential synergistic or cooperative effect. And these were what we call the second generation compounds. So we found that these compounds had, like number 1, they had anti-cancer activity, but when exploring about the stability, and a little bit as expected, we were losing the cyclopentadienyl rings that carry the gold. So the molecules couldn't be intact to reach the tumor. So that was defeating the purpose of having the 2 fragments into 1 molecule. So we quickly switched to what we call the first generation, in which we wouldn't have the gold derivatives attached to the cyclopentadienyl, but instead we had carboxylates that would bind nicely to the titanium and would be more stable, those bonds, than the titanium carbon bonds. And these first generation derivatives we did with different, with phosphines. So we will have the carboxylate and then we will have a phosphine attached to the gold. And finally, we move to second generation, those compounds in the scheme that are called titanocref and titanofin, which have a carboxylate and a thiolate, and then that gold can be attached to the thiolate, which is something gold 1 really likes to do. And then a step forward is to substitute that phosphine by heterocyclic carbines. So we have the different generations of compounds that we explore. So I'm gonna talk about the second generation that contain phosphines, because these were the lead compounds, the one that we really saw were more beneficial, and we did further work with them. So the synthesis is not, it doesn't come from titanosin dichloride, but from titanosin dimethyl. This molecule is unstable at air, so you have to handle it under argon, and you have to do that synthesis. But it's really nice. In less than 1 hour, you can coordinate this derivative with a carboxylic acid to the titanium. And despite of the different stoichiometries that we try, we always got the derivative that was bimetallic with 1 titanium and 1 gold and still 1 methyl. The most surprising thing for us was that this derivative was air stable for the most part, which is pretty unique when you talk about these derivatives. And the compound was stable in DMSO solution for at least 72 hours. And when they decompose, we could follow the patterns of the composition. So for the most part, we were pretty satisfied with the stability of these compounds to move to biological experiments. So we explore cell viability, and this is only a summary because we try many different renal cancer cell lines and also non-cancerous cell lines from patients like RCPT. So here we have the different compounds, and as I said before, cell viability, IC50s, is a type of colorimetric assay in which you are gonna determine the concentration that your compound is able to kill 50% of the cells, okay? So the lower this value, the better it's gonna be. You usually move in a range of micromolar, and anything below 10 micromolar starts being interesting. And if you go below a micromolar, if you go to the nanomolar, it becomes really interesting. So we explore CACI1 cells, which are clear renal clear cell carcinoma, which is highly metastatic, and we compare with non-cancerous cells that are fibroblasts called IMR90. So you can see here the different compounds, and when you explore the activity of heterometallic compounds, you are gonna see, you're gonna study the activity of the heterometallic compound, but you can also, you need to also see the fragments, the metallic fragments, in this case, the metallic fragments of gold that were CREF and FIN. All these names were given by my PhD student, Tina Eli, which is on the screen. She came with these names, and that's how we patented these compounds. And then also the titanosin dichloride, which is the other fragment. And then you also want to do some studies on synergism, or the gold and the titanium together, not on the same molecule. And as you can see, for the heterometallic compounds, titanocref and titanofin, they were in the nanomolar range in renal cancer, and with quite good selectivity, especially titanocref was very selective. And what we found is that the combination was not good. So just simply putting the titanosin dichloride and the gold was not good. And that was a big thing for us to move forward. In addition, these derivatives didn't show interaction with DNA, which was different from the first generation and different of what titanosin dichloride does. But they didn't display interaction with DNA, with plasmid DNA, or when we use calthymus DNA. So one of the things we did was cellular uptake. So you can use techniques as mass spectrometry, ICP-MS, or you can also use adsorption, atomic adsorption spectroscopy in order to account for how much metal. So this is an advantage in general of metallic based compounds, that you can see how much metal there is. And what we found is that the compounds, the cellular uptake of the heterometallic compound, both metals were uptaked almost identically, which told us that at least in CACI cells, they co-localize, and possibly the compound was still robust and intact when they entered for the most part. The parts that were linking both were there. We study the cell death, and there are different things in this slide. So you usually study if the compounds die by necrosis or by apoptosis, which is like the suicide of cancer cells, and is very much liked that the compounds die by apoptosis. And on the left, with those quadrants, you have that type of experiments. And for those experiments that you do by flow cytometry and also labeling with a dye, we saw that our derivatives were indeed highly apoptotic, and as apoptotic or even a little more than auranofin. And then we went to explore the type of cell cycle. So the cells all have a cycle by which they die. And you all usually compare with cisplatin and some others. In this case, we compare with auranofin. And what we saw for our heterometallic compounds, titanocref and titanofin, is that the cell death cycle was mostly in the phases G2, M, and S. And when you look at auranofin, it goes to G1, G0. And this is telling us that the titanosin fragment is having an influence, because it really modifies the phase of cell death. So the titanosin fragment, despite it's not very cytotoxic, as we saw before, it still has a huge impact in the cell cycle. So far, so good?
Yes, thank you.
Okay. Okay, so the next thing we did was, and everything in these studies, we took one particular cancer cell line, renal, khaki, which is highly metastatic. So how we can see if our compounds can potentially inhibit metastasis? You cannot see that a metastatic model should be in animals, but in cells, what you can see if is the compounds inhibit migration or invasion. So in order to see if they inhibit migration, you use what is called a two-dimensional experiment or assay, which is called cell migration inhibition assay, or a scratch assay, and then you have your plate, which is full of cancer cells that would be in the line of the DMSO. You will have your plate with cancer cells there, and you do a scratch or injury. So you separate, so the cancer cells get separated. And when you allow that plate with cells to grow, that little scratch is going to be closing down because the cells are migrating. So when you treat these plates with your compounds, you want to see if that gap closes down and then there is no inhibition of migration, or if that gap keeps up or even increases, right? And this is what we saw, that our derivatives, titanocref and titanofin, were quite good in inhibiting the, in still having it expanded that gap, right? Better than the gold derivatives, cren and fin, and almost as good as auranofin, that is very well known to inhibit migration. So we also moved to a tridimensional assay invasion, which is done in a matrix gel and basically observes how if the areas in which these cells are invading and how they prevent. So when you look at the picture of the MSO, you see all these aggregates and cells, right? And when you look at the one of auranofin, which is really good in prevention of inhibition, you see much less cells. And we saw that it was both heterometallic compounds were good in inhibiting this invasion, especially the derivative that contain the triethylphosphine fragment, like auranofin. So these compounds inhibit migration and invasion in vitro, and that's a sign that they can inhibit potentially metastasis. So not only they are cytotoxic, but they also could potentially inhibit metastasis. Another effect that you would like to explore is if the compounds inhibit angiogenesis. So usually cancer cells have vasculature around, and through that they can feed. They need vasculature so they can get nutrients and then feed and then become bigger. So if you have compounds that are inhibiting that vasculature around the tumor, you are preventing the tumor to grow and to and it can be potentially eliminated. So this is very important to find compounds that can inhibit this process, this angiogenesis. And the way you do this experiment, you use some cells that are human umbilical vein endothelial cells. So they are vascular cells, not cancer cells, vascular cells. And then you see them in a plate, and you see if those vessels are disrupted. You can measure, you can be measuring the tube nodes of the vessel and the tube length to see if they get smaller. And when you see the picture of the MSO, especially after 24 hours, you can see the formation of those vessels, right? And you see how when you add compounds, this can be very much disrupt. And all these compounds disrupted this vasculature, so they inhibit angiogenesis, which is good. Now we know that they are cytotoxic, they are selective, they inhibit migration and invasion and angiogenesis. So what we did next was to explore if they inhibited molecules that are related to these processes. So we did proteomic analysis, and then we were into different details. This is quite a bit of biochemistry. I'm not going to get into all that. But what we found that was very important is that the compounds inhibited thioredoxin reductase. So auranofin in blue in the scheme A, auranofin in blue, is known to be one of the best thioredoxin reductase inhibitors there is. Okay, so our compounds in red and purple really inhibited thioredoxin reductase. And this is an enzyme that is related to resistance to cisplatin. So it's really good that the compounds can inhibit the thioredoxin reductase. It also helps the overexpression, helps the cell to evade apoptosis, which is not good. We also look to see if the compounds inhibited what is called VEGF, which is vascular endothelial factor, which is related to the angiogenesis effects that we saw earlier. And again, we saw a reduction or inhibition. And then we were checking into interleukins or the tumor necrosis factor and others, and metalloproteases, and all of them are associated with the processes I said before. So from all these inhibition assays, we could confirm that the compounds inhibited angiogenesis, migration, and invasion because they inhibited certain and specific enzymes that are associated to these processes. And lastly, the last thing we've done in terms of in vitro with these compounds was done by our collaborator at the University of Hawaii, who is now at the University of Louisiana. He did a microanalysis of mRNA. So in order to see the genes that are altered when you treat CACI1 cells with titanocref, and then there is all these different genes that were associated and identified, etc. And what I can tell you, like as a summary, is that this titanocref alters apoptosis and alters pathways that are related to reactive oxygen species, which is not surprising, being that they seem to accumulate in mitochondria as well. But they are very, very effective because even in nanomolar amounts, they can affect these pathways in 3 hours. And it was clear that there is an activation by reactive oxygen species of the apoptotic pathways. So we did in vivo studies, and I must say that we did 2 in vivo studies. We did one in vivo study in 2015, which was our first one, with titanocref. We hadn't designed titanofin to be closer to auranofin at the time. And when we did the study of titanocref, it was good. We saw tumor size decrease of 50%, but the pharmacokinetics analysis was not good. We were dosing every other way and every other day, and that was not good. We needed to do things in a different way, and I'm gonna walk you a little bit. The first thing you do when you do in vivo studies, and you wanna do in vivo studies always in mice that are bearing the tumors, is you are gonna have the mice, and the mice have an immune system that has been suppressed, okay, and you are gonna inject human cancer cells, in this case is CACI1, in order to generate tumors. So you want to give tumors to the mice, the immune system is suppressed, so they can grow because it's human, right? And then you want to do the pharmacological experiment with this type of mice. So we started, first of all, getting the lethal dose. And I know it's very sad, and for those of us who really love animals, you know, to have mice that are going to be sacrificed in order to see how higher you can get to the lethal dose. That's why we don't do many in vivo experiments, and only for compounds that are really promising. And the in vivo protocols we have to file are really something else. It's not easy. They don't make it easy. So there is a lot of good things happening around for the mice so they can be treated humanly, okay? And when we did this, we took 5 different mice for each group, and then we injected these compounds in different doses in order to see the dose at which the mice end up dying, right? And this was a dose of 30 milligrams per kilo for titanocref and 45 milligrams per kilo for titanofin. Just to put this a little bit into context, in the 90s, there was a lot of research done with gold phosphate derivatives that was abandoned because it was very, very toxic. So these compounds had lethal doses of 1 milligram per kilo. So it's good that the lethal doses of these compounds are a little bit higher. So the next step with different mice is to find the maximum tolerated dose. So not the dose at which the mice die, but the maximum dose in which the mice still behave properly, in which they groom, they eat, they move properly, and also that they don't lose much weight, because you don't want cancer patients to lose. Usually the limit is 8% of weight loss that is acceptable. And we explore with groups of 5 mice again and different doses. And then we found out that the doses had to be below 15 milligrams per kilo for the titanocref that has the triphenylphosphine, and 30 milligrams per kilo for the compound with the triethylphosphine, which is also present in auranofin. I think it's important to notice this because it seems there is an advantage to have triethylphosphine as a ligand for the gold. So once we had this, and we did a pharmacological study, which I'm gonna have to come here. So we did the pharmacological study in 72 hours in different groups of mice. And from here, we got blood work, and we had obtained half-lives. And then we learned that we had to do these experiments and dose the mice every 3 days, because every 2 days was too much. The drug couldn't leave the system. Okay, this we learned from the pharmacokinetic analysis. So we did what is called the in vivo efficacy trial. You get different groups of mice that have tumors. The tumors have to be at least 300, 400 cubic millimeters of size. You have to have a minimum size, because you are going to start dosing the mice either with vehicle, which is in this case is just water with DMSO, or else with your compounds, okay? And then you are going to be measuring in a blinded study the tumor size. So we could see that our control mice with only a little bit of DMSO, the tumors grow and grow, right? And in the mice treated with titanocref and titanofin, not only the tumors don't grow, but also the tumor size decrease, okay, which is different. And the doses are given by intraperitoneal injection. And this study was in 21 days, okay? And the groups were of 6 mice. So you want to have mice that are mixed male-female, unless it's a cancer that is mostly female, like breast cancer or ovarian cancer, or prostate cancer for male. But nowadays they want to have a mixture of female and male mice. Any questions about in vivo? A little bit that you may have?
How much is the concentrations for you using for all that not toxic for the human?
How much is the concentration for the humans?
Disculpe, es que my English very rusty.
Yes, you can ask in Spanish if you want.
Okay. ¿Cuánto es la concentración mínima que se tiene que utilizar de oro para que esta no sea tóxica para los tejidos o para las células sanas del cuerpo?
¿De ratones o de humanos?
Para ambos, si me lo podría decir.
Pues la verdad es que eso no se sabe de antemano. En principio, eso lo vemos después. Lo que hicimos fue tener una cantidad en la que aquí teníamos, ¿te acuerdas? La cantidad en la que mata es de 30 o de 45, y la cantidad que pensamos como maximum tolerated dose era de 15 o de 30. La cantidad que nosotros empleamos para este estudio in vivo fue de 5 miligramos y de 10 miligramos. Bajamos 3 veces el maximum tolerated dose. So we decrease 3 times the maximum tolerated dose for the in vivo efficacy trials. But
Thank you.
there is not an amount of gold. Every compound is different. Does it make sense? So
Yeah,
we
sí, me
knew
hace-
that they were behaving, so at 15 milligrams per kilo of titanocref or at 30 milligrams per kilo of titanofin, the mice didn't didn't have a weight loss or anything like that, were behaving nicely. And with 3 times less, we did this in vivo efficacy trial and we saw this decrease of tumors, right?
Yeah,
Now what you are
thank you.
asking, yes, now let me continue because I'm gonna reply to your question. So we saw this tumor size decrease of 51% and 60%, which was great. It's very high, this is not very normal with chemotherapeutics. So another thing that we did before I go to the histopathology, Oscar, it was to see if in the tumor tissue, if we could see if there was apoptosis and also if it was a decrease of cell proliferation. So you can add dyes, to these tissues, and indeed we saw the green one is cell proliferation marker, how it decreases in the mice treated with titanocref and titanofin, and how the apoptosis marker, which is caspase 3, how it increases. So we could see that the tumor size that we saw in vivo, it goes with decrease in cell proliferation and an increase of apoptosis. We also wanted to see effects in angiogenesis, right, for the mice, and to see if there was a disruption in the vasculature around the tumor. And the way you do this, you have to inject the mice on the tail with different dyes. One dye is lectin, and which is going to be giving you a measure of the vascular wall. And another dye is a sugar, the stram vascular likeness. So in the mice which has not been treated with gold, you can see how the vasculature is still almost intact. And you see this because there is a lot of red and a lot of green, right? Whereas the mice treated with titanocref or titanofin, you basically don't see the green or the red, especially the one for titanofin. So this means that there is an in vivo inhibition of angiogenesis. Now, we haven't done yet a mice model to evaluate metastasis because you really, in order to do that, the mice need to have mice cancer, not human cancer, mice cancer, and this is an next step. And in order to do that, you need to have the mice with immune system, not immunocompromised, and that would be the next step. Can we also stop metastasis in vivo? But we saw that we produce apoptosis in vivo, and we inhibit angiogenesis in vivo, and we decrease tumor size. Now, I'm going to go a little bit about the toxicology, which is what it was worrying Oscar, rightly so. So when we did the pharmacokinetic studies, in addition to the blood markers that were all quite okay, we wanted to see the amounts of metals and where they went. So we did different organs, and these are the most important: liver, kidney, and tumor. And in the beginning, when I saw this, I was very preoccupied because there was more in the liver and then the kidney than in the tumor for the most part. In titanocref, there is more in the tumor. And I thought, oh, it's very little in the tumor. But our collaborator, pharmacologist, Dr. Swayan Prabha, told me, no, this is a lot for the tumor, for a chemotherapeutic material. This is good. And it's good, it's going to the liver and not to the kidney. So this is quite okay. This is a preliminary pharmacokinetic analysis done in only 72 hours. So you see, one thing that you see is that you have the same content, and again, the same is a little bit between brackets, is pretty much the same between gold and titanium. It accumulates the same in the liver, the kidney, the tumor, right? When you do this after you have finished the in vivo trial, you also have a biodistribution into liver kidney tumor, but you can see much more accumulation in tumor, but less accumulation in liver and kidney. And this particular pharmacokinetic profile, according to pharmacologists, is good. And one of the things that it allows us to see is that these moieties are robust because there is co-localization of titanium and gold. And again, it's not the whole molecule, it's the part of the molecule that connects the titanium with the gold, which is relevant. So now when you do these in vivo efficacy trials and you kill the mice, you get tissues not only for doing those mechanistic experiments, but you also get tissues from 48 organs, different ones. And again, this is only some of the most characteristics, which is the liver, the spleen, and the kidney. And you want to see this along with blood work to see how different is the mice that were not treated from the mice that have been treated with the compounds. And what you can see in this picture is that there is no difference between them. And also there is no difference in the body weight of these main organs and other organs. Okay, so that was really interesting for us and really hopeful because the metal tissue accumulation that we observed before in some organs, it has no pathological consequence. Okay, the liver, despite accumulating the titanium and the gold, it didn't have a pathological consequence other than elimination of the cancer, right? So this is where we are right now. And we did some other studies in which we have been following what happens to these compounds in plasma. And we know that at least 70% of titanocref remains in plasma after 60 minutes, which is good. So these compounds are very robust, even if they lose the cyclopentadienyl, which is what happens, and they travel and possibly binding, transferring, and then going to where they need to go. Did this clarify, Oscar? It's not that you can say how many milligrams of gold. I mean, one thing is metallic gold and another is gold that is inside, I mean, like is part of a compound, which has a different solubility. But you need to do these studies after to see how toxic they were. Did it clarify?
Muchísimas gracias.
Okay. Okay, so I'm gonna move, and this is gonna be faster because I wanted to explain to you the experiments a little bit. So we were also interested in adding to gold ruthenium parasimine derivatives because they are very well known as anti-metastatic agents. And we first designed these compounds that contain a phosphine between the gold and the ruthenium. This is ruthenium 2. And we had a simple compound with chloride and then with a thiolate. And then we saw that again, the compounds had a synergistic effect. It was much better than the cancer activity of one molecule. We saw high cytotoxicity against colon and pancreatic cancer. And then we also observed they didn't interact with DNA. And we saw a little bit of inhibition of cathepsin B, which is associated to anti-metastatic properties. But these compounds were neutral and we were not completely satisfied. We also wanted to incorporate N-heterocyclic carbines. And then we came up with this synthesis, which my postdoc at the time, Jacob, didn't want to do. He didn't want to do this. I had to go to the lab in 2015 and do this reaction myself to convince him, which I think is one of the latest times I've been doing the synthesis myself. So you can start by a simple gold heterocyclic carbene, which is neutral, and chloride, and you can use a silver salt to remove that chloride and generate an intermediate in which the perchlorate of the triflate are coordinated to the gold. These are anions, but they can also coordinate covalently. And then you can add a ligand, a neutral ligand, and then you generate this gold ligand neutral, heterocyclic carbene neutral, and then you have an anion outside. And this is true for phosphines, arsines, etc. And then what we did was adding as a ligand the ruthenium parasimane with 2 chlorides and the DPBM. And then we generated these compounds that were cationic, and we thought they were going to be better and with different heterocyclic carbenes. And I only did one reaction. Then Jacob was immediately convinced, and he did a wonderful job. And I must say that he came up with the synthesis of the carboxylate thiolate, that the optimization was him. So yes. Anyhow, so we chose from the different ones we did, we chose this one that again, Tina named as Rancy. And of course, you have to do all these experiments and having controls, which is R is the compound of ruthenium and C is the compound of gold with the carbene. And this is what she did with 2 undergraduate students. And what they found is that the compound was highly cytotoxic and selective in khaki as well, that it inhibits thioredoxin reductase, as we had explained before. And also they were able to demonstrate strong anti-migratory properties that also could be explained by the inhibition observed of different metalloproteases, cathepsins that are present in khaki cells. And they also observe inhibition of angiogenesis for these derivatives. It seems that the gold really drives quite a bit in terms of the angiogenesis inhibition for both the titanosine derivatives and the ruthenium gold derivatives. We did in vivo again, and Oscar again, we did everything. The maximum tolerated dose, which in this case was 35 milligrams per kilo. The half-life was 40 hours, etc. And in this case, what you can see here is that the tumor, the mice treated with the vehicle, the tumor grows, and the mice treated with the Rancy, it didn't grow. It didn't decrease the size, which is very unique, but it prevented the growth inhibition 100%, which is what you want from a chemotherapeutic. So it was good. Not as good as the titanosines, but it was good. And again, we did all the blood work and the histopathology, and we saw the same, no signs of pathological complications as a result of the treatment. So it seems to us that there is a lot of demonization of metal-based drugs, that everything is like cisplatin and everything is toxic, but it isn't. It depends on the compounds, it depends on the oxidation state, on the ligands. And we also here were able to demonstrate tumor, that it was the tumor growth inhibition had also increase in apoptosis and decrease in tumor cell proliferation. But we didn't see inhibition of angiogenesis in vivo this time, which was surprising because in vitro, the inhibition of angiogenesis for this derivative was higher than the one for the titanosines, but the titanosines were better in vivo. For the most part, for these projects, we've been able to translate in vitro almost to in vivo, but it's not always the case. And there are little things that don't translate equally well from in vitro to in vivo. So this is also how in vitro that inhibition of tumor growth coincided with the inhibition of specific markers, etc. So, okay. And lastly, I'm gonna talk about different set of compounds that more recently we've been involved, which is with platinum-4 anti-cancer agents. As I said in the introduction, there are different strategies. So platinum-2 compounds, they are not very bioavailable, only 5% usually go into cell and in the nuclei, etc. So one of the strategies is to have a much more inert disease species, because platinum-2 is D8 species. And so when you have a more inert disease species, platinum-4, also with a metal that can be reduced from platinum-4 to platinum-2, inside the cells, but different molecules, and this is being demonstrated that platinum-4 compounds can be reduced into the cells, you are going to free your platinum-2 and you are going to free ligands, right? But this is a strategy so these compounds can reach the tumors and then get reduced inside the tumors. And if you rationally design it, your ligands can also be bioactive. And why not? They can contain a second metal, okay? Which is what we did. We wanted to have a second metal, our gold compounds, and this is the work of PhD student Javier López Hernández. So we really capitalize on our compounds from before, from the first generation that were not so good, but in this case, they were good for us with carboxylate and a phosphane. And then we just modify and activated this ester. And then you can do a reaction with the OHs from oxoplatin, which is the platinum-4 form of cisplatin. And then we were able to get this first compound with 1 platinum and 1 gold. We also saw that by using coupling reagents like TBTU and some others and a base, we don't need to activate the ester. And then we can have compounds that contain 2 golds and 1 platinum. And we demonstrated this with platinum-4 derivative of cisplatin, oxoplatin, or with the platinum-4 derivative of oxaliplatin. And we characterized all these compounds and we evaluated the stability, etc. The only thing we couldn't do was to get crystal structures despite of many, many attempts. So we asked our collaborator, Dr. Jose Cerón Carrasco, and he's been doing DFT calculations for us. So we have an idea of the structure of these compounds that have been confirmed with experimental infrared and diffuse reflectance spectroscopy. And it also has given us an idea of the stability of these compounds towards reduction and whatnot. So we have explored, because we don't know where they are going to be active or not, we took a panel of different cancer cell lines. So MDA-MB231 is a triple negative breast cancer cell line. SKOV3 and A2780 are ovarian cancer cell lines. TACI is renal and P24 is bladder. IMR90 is the fibroblast, not cancerous. And we did all these comparisons of IC50 values, 72 hours, and comparing with cisplatin, oxaliplatin, and the derivatives of platinum-4. The derivatives of platinum-4 are much less cytotoxic than the derivatives of platinum-2. This is normal. Still, they are more selective than cisplatin and oxaliplatin. The selectivity is better for ovarian, but in the other cancerous cell lines, they are not so good, these derivatives of platinum-4. So what you can see from here is our derivatives incorporating platinum and gold. They are highly cytotoxic. In ovarian, not as cytotoxic, that's cisplatin, which was to be expected, but they are more selective. And we were really interested when we saw this derivative that contains the oxaliplatin core, because it was quite cytotoxic to triple negative breast cancer while being quite selective. And in my laboratory, we explored triple negative breast cancer quite a bit with ruthenium compounds. So we decided to take this cell line and explore it a little bit further. So one of the things that we saw was if the compounds were apoptotic. We took a particular compound, this compound 5, and is apoptotic, a little bit less than the gold derivative, but apoptotic after all. Then in terms of the cellular and organelle uptake, we saw that the uptake again, the platinum and the gold are uptaked at the same time. And in terms of the organelle, we saw that the compound for both gold and platinum, it accumulates more in the nucleus and the mitochondria, but mostly on the nucleus. So you will ask me if these compounds interact with DNA. We've done many studies with plasmid, DNA, calthymus, with and without reduction. And for the most part, it doesn't seem that they interact with DNA. So we are also exploring the inhibition of thioredoxin reductase at the moment to understand the mechanism a little bit more, and we will explore DNA quadruplexes. So we use 3D spheroids of a particular cell line in triple negative breast cancer. And usually with cisplatin, what happens is that it has good activity in monolayers, but not so good in 3D spheroids. But in this case, it seems that it's very similar, which is very encouraging in order to go towards in vivo studies in mice. And then we observed that this derivative, the platinum with 2 golds, also inhibits migration like crazy. This is one of the best inhibitors of migration we have observed in our lab. And then in terms of the inhibition of angiogenesis, it does, but surprisingly, the heterometallic compound is a little worse than the gold and worse than the platinum derivatives. So this is where we are, and I think in terms of the conclusions, I hope that I have shown you how 2 metals are indeed better than one when chosen rationally, right? And how we've been able to confirm co-localization of both metals inside cells. So it tells you that at least the biological active component that contains both metals is still intact. We have observed that ruthenium and gold and possibly platinum and gold, there is a synergistic effect. So both metals separately have an effect. But we believe that in terms of the titanium gold, the second metal is acting as a modulator because it's not very cytotoxic, etc. So we believe it's more of a cooperative effect. It improves the physicochemical properties of the molecule quite a bit. And then we have demonstrated the potential of these families of compounds as chemotherapeutics in renal cancer. And we have to see, we are really now looking into ovarian and triple negative breast cancer for the platinum-4 gold compounds. So what are we going to do next? We are going to continue validating all the platinum gold. We have a lot of work in front of us. We are currently, a company is currently doing for us cell-based painting assays. So it's going to tell us where the compounds go, which is going to help with mechanistic studies. We're going to be working with organoids derived from real patients from different ethnic backgrounds, and we are going to put them in mice for all lead compounds. And we work quite a bit on targeting vehicles and encapsulating materials. And of course, once we select one compound, we are going to go scale up and improve formulations. So I want to thank everybody who were involved in this project. The people in red are still around because they are not graduated as yet, or they are professors I collaborate with. I collaborate also with ISRC Memorial, but I haven't spoken about this. And of course, thank you to the money I got to do all these projects. And I just want to show you where Brooklyn is in New York City. Is one of the 5 boroughs. It's the coolest borough, okay? I know that Arthur likes Queens, but I like Brooklyn, which is the coolest city on the planet. And it has many attractions. It has a beach that we go to. It has several beaches, Brooklyn, by the way, and it's quite nice. And we have our own Prospect Park, which is great as well. And this is a little bit of Brooklyn College in the different seasons. We have all seasons here. So thank you very much.
Round of applause for Dr. Gonzalez. Thank you so much for that. Very insightful, and it really expands the set of work. Does anyone have any further questions with regards to the research that was discussed today?
Hello. Yeah. I have a quick question. Great talk, Professor. I was wondering, have you, like you were working with bimetallic complexes, have you tried like doing only dinuclear to see like like the difference between just using the dinuclear, the same metal centers, and then comparing it to the bimetallic ones?
Yeah, not for this work, but in the past we did a little bit of that as antimicrobials. And we didn't see, I mean, there is always a little bit of effect when you have two of the same, but for this particular project, we really wanted to add one of the things we want to overcome is resistance to treatment. That's what we thought, having metals with different mechanisms of action was going to be more interesting. But yeah, there is an improvement with polynuclear complexes. There is a lot of work done by Nicholas Farrell on this and platinum.
Thank you.
I have a question. Nice talk, Maria. Can you study like electrochemical properties of these complexes, like through cyclic voltammetry, the redox and reduction reactions of them?
Yes, you could. We did with some ferrocene derivatives that we study. I'm not an electrochemist myself, so I think we haven't done more just out of lack of expertise. But you can do some for certain. You can do study the electrochemical properties, and that would be something very interesting, especially when you talk about reductions inside the cell. So that's of interest. Yes.
Okay. And another question, so I see the heterocyclic, basically the extension that you give, right? The derivatives. Have you tried any type of making other derivatives or adding functional groups to that cycle?
Cycle, you mean the heterocyclic carbons?
The heterocyclic carbons, yeah.
Yes, we did quite a bit of functionalization, but after we saw the efficacy in vivo, we sort of didn't go further, you know, like once we see a lead that is good enough. I mean, you can do things, but then there is not so much to justify continuing and doing much more in vivo. But there is always different modifications you can do. I guess we are more now towards mechanisms that other improvements synthetically, but yet for certain, I'm pretty sure that you can fine tune even more.
All right, cool. Thank you. A very nice research. I like it.
All right, so another round of applause for our guest speaker. Maria, if you have a moment, I would like to continue to speak to you for a moment.
And I apologize, I didn't realize I was taking so long. I guess I went too much into explaining the assays, but I thought it could be interesting for students. So apologies.
It was very helpful. No, you did very well. Thank you. All right,
Bye-bye,
everyone. I'll see you
everyone.
later.
Thank you.
Adios.
Bye-bye.
Gracias.
Vemo.